Hello, my name is Joshua Mugabi. I welcome you to Genetic Engineering. What is biotechnology? Biotechnology is the application of biological knowledge to obtain new techniques, materials, and compounds of pharmaceutical, medical, agrarian, industrial, and scientific use. For example, the practical use. The pioneer field of biotechnology were agriculture and the food industry. But nowadays, many other practical fields use its techniques. What is genetic engineering? Genetic engineering is the use of genetic knowledge to artificially manipulate genes. It's one of the fields of biotechnology. At the present level of the biotechnology, what are the main techniques of genetic engineering? The main techniques of genetic engineering today are recombinant DNA technology, also called genetic engineering itself, in which pieces of genes from an organism are inserted into the genetic material of another organism, producing recombinant beings. The nucleus transplantation technology, popularly known as cloning, in which the nucleus of a cell is grafted into a nucleated egg cell of the same species to create a genetic copy of the donor individual. The technology of DNA amplification, or PCR, polymerase chain reaction, that allows million replications of chosen fragments of the DNA molecule. The recombinant DNA technology is used to create transgenic organisms like the mutant insulin-producing bacteria. The nucleus transplantation technology is in its initial development, but it is the basis, for example, of the creation of Doll, the sheep. PCR has numerous practical uses, as in medical tests to detect microorganisms present in blood and tissues, DNA fingerprints, obtainment of DNA samples for research. What are restriction enzymes? How do these enzymes participate in the recombinant DNA technology? Restriction enzyme or restriction endonucleases. These are enzymes specialized in cutting the DNA fragments, each acting upon specific site of the DNA molecule. Restriction enzymes are used in recombinant DNA technology to obtain with precise pieces of DNA molecules to be later inserted into another DNA molecule cut by the same enzymes. What are DNA ligases? How do these enzymes participate in recombinant DNA technology? DNA ligases, these are enzymes specialized in tying the complementary DNA chains to form the DNA double helix. These enzymes are used in recombinant DNA technology to insert pieces of DNA cut by restriction enzymes into another DNA molecule, submitted to the action of the same endonucleases. What are plasmids? Plasmids, these are circular DNA molecules present in the genetic material of some bacteria. They contain genes responsible for bacterial resistance to some antibiotics and for proteins that cause virulence, pathogenic hostility. How is genetic engineering used to create bacteria capable of producing human insulin? In the production of human insulin by bacteria, the human insulin gene is incorporated into the genetic material of this microorganism. The mutant bacteria multiply, forming a lineage of insulin-producing bacteria. In bacteria, there are circular strands of DNA called plasmids, microsomes, that act as accessory to the main DNA. To create a mutant bacteria capable of producing insulin, a plasmid is inserted to the action of restriction enzymes, specialized in cutting DNA fragments. The once circular plasmid is then opened by restriction enzyme. The enzyme is used to cut the human DNA molecule containing the insulin gene. The piece of human DNA containing the insulin gene then has its extremities bound to the plasmid with the help of DNA ligases. The recombinant plasmid containing the human insulin gene is then inserted into the bacteria. Another human hormone already produced by recombinant bacteria is DH, or somatotrophic or growth hormone. The insertion of DNA molecule into cells of individuals is also a method of gene therapy, promising treatment for genetic diseases. In gene therapy, cells from the organism deficient in the production of a given protein receive, by means of vector or virus, pieces of DNA containing the protein gene, and then they begin to synthesize the protein. What is cloning? Cloning is the making of an organism genetically identical to another by means of genetic engineering. The basis of cloning is nucleus transportation technology. A nucleus of a cell is extracted from an embryonic cell, and this nucleus is inserted into the previously enucleated reproductive cell. The egg is then implanted in the organ where the embryonic development will take place. If embryonic development occurs, the new organism will have identical genetic patrimony to the organism owner of the cell, whose nucleus was used in the transplantation. What is PCR? How does PCR works? PCR, or polymerase chain reaction, is a method to synthesize many copies of specific regions of a DNA molecule known as the target regions. Its inventor, Carey Mullis, won a Nobel Prize for Chemistry in 1993. First, DNA to be tested is heated to cause the double helix to rupture and the polynucleotide chains to be exposed. Then small synthetic sequence of DNA known as primers and containing nucleotide sequence similar to the sequence of the extremities of the region of this to be studied. For example, region containing a known gene exclusive of a given organism are added. The primers paired with the original DNA in the extremities of the gene to be amplified. Enzyme known as polymerases that catalyze DNA replication and nucleotide supply are added. Primers then are completed and the chosen region is replicated. In the presence of more primers, more nucleotide millions of copies of that specific region are generated. PCR is very sensitive, even using minimal amount of DNA. What is the fact of molecular biology on which DNA fingerprint is based. DNA fingerprint, the method of individual identification using DNA, is based on the fact that DNA of every individual contain nucleotide sequences exclusive to each individual. Although normal individuals of the same species have the same genes in their chromosomes, each individual has different alleles, and even in the inactive portion of the chromosomes, heterochromatin, there are differences in the nucleotide sequence among individuals. Why are the recombinant DNA technology and nucleus transplantation technology still dangerous? The recombinant DNA technology and nucleus transplantation technology, or cloning, are extremely dangerous since they are able to modify in a very short time the ecological balance that evolution has taken millions of years to create on the planet. During the evolutionary process, under the slow and gradual action of mutation, genetic recombinations, and natural selections, species emerged and were modified, and genetic patrimonies were formed. With genetic engineering, however, humans can mix and modify genes, making changes of unpredictable long-term consequences, risking creating new plants and animal diseases, new types of cancers, new disease outbreaks. It is a field as potentially dangerous as the manipulation of nuclear energy. What is the main moral problem about cloning human individuals? Beside biological perils, very serious moral problem involves nucleus transplantation technology concerning humans. An individual right of human being is offended when man or woman is made as a copy of another. Since it is impossible to first ask if the person to be generated wants or not to be generated genetic copy of another person, certainly the most important human rights is being offended. One is individual freedom. When human being is obliged to be genetic copy of another, it is indeed danger to democracy, whose most basic principle must be non-violation of individual freedom. Thank you very much for participating in genetic engineering. Please do like, share, and subscribe.